So globally, an estimated 3 billion people rely on rudimentary open-fire cook stoves to cook food, boil water, and heat their homes. The problem is that these cook stoves emit a significant amount of smoke and indoor air pollution, whose inhalation has been linked to deadly diseases of the respiratory tract and lungs. The World Health Organization reported that in 2012, cook stove-induced indoor air pollution was responsible for 4.3 million premature deaths, almost all of them occurring in developing countries. Now, there have been efforts over the past several decades to implement technologically improved cook stoves designed to use less fuel and produce less smoke. And although these cook stoves have been successful in laboratory settings, they've often failed to be effective in field studies, illustrated by low rates of long-term adoption owing to poor maintenance of the stoves. Like a lot of other examples of failed technology in global health, it's a problem of sustainability. So together with some colleagues at Nyaya Health, which is an organization that runs a health system in far western Nepal, what we're attempting to do is to take a look at this issue from a different perspective by framing the access to clean air as an integral component of the right to health. The first part of my project involved advocating for this rights-based approach to indoor air pollution. I wrote a piece, was able to get it published, and in it I advocated for improved cook stove programs to be integrated into public sector healthcare delivery systems. The second part of my project involved actually traveling to Nepal to first collect some baseline data on the prevalence of respiratory illness and cook stove usage, and second, to do an initial assessment of the readiness of the healthcare system to incorporate improved cook stoves programs into routine primary care. Now, I am a human physiology major, interested in pursuing a career in medicine and public health. And so my interest in this work really came from a desire to better understand how health systems are developed. My goal was to use this issue of indoor air pollution as a vehicle into understanding the true potential of the primary care system and the processes involved in realizing the right to health. Thank you.
In 1926, James Laver, an English writer and curator at the Victoria and Albert Museum, wrote a poem titled The Women of 1926. This poem explored the new music, dancing, and other aspects of modern life in the 1920s. Most significantly, Laver describes the distinct fashion of the 1920s in 3 lines which read, "Our waists are round about the hips, our skirts are well above the knee, we've boyish busts and eaten crops." The poem continues, but these lines accurately describe the popularity of shorter skirts, androgynous dresses, and short hairstyles in the 1920s. Further, all of these styles could be achieved in new ready-to-wear forms and in materials such as rayon, which was made possible as a result of streamlined mass manufacturing technology. These changes in style and production lead fashion historians to cite a democratization of fashion, in which dress was no longer a key indicator of class as a fundamental characteristic of fashion in the 1920s. This change in fashion mirrored changes in British society as a whole. Beginning in the late 19th century, historians claim a decline in the British elite's political, economic, and social power. However, London stores continued to use a relationship with the elite in their promotional material in the 1920s. Even with the relative democratization, fashion functions through constant change to prompt anxiety of not having the most up-to-date look. Thus, retailers of fashion, such as department stores, need to incite anxiety in their consumers in order to sell goods. My project examines the use of the elite in the promotions for 3 central London department stores: Liberties, Selfridges, and Harrods. By the 1920s, these stores were well established as important shopping centers in London. Each of these department stores continued to use the elite in various ways to promote their store's fashion departments in the 1920s, offering a counter-argument to the notion that the elite had all but disappeared, even as arbiters of fashion. The advertisements, events, and design of these stores suggest a continued saliency of the elite's social power in British society and at a time when it was supposedly in decline.
As both a musician and a scientist, I've always been fascinated by the ways in which music can affect a listener, whether it be cognitively, psychologically, physiologically, or even biologically. I'm also intrigued by the different levels on which music has these effects. Not only is music commonly used interpersonally as a means through which people connect, but it also has great intrapersonal worth to us as it impacts us each on an individual level. These initial ideas led me to integrate my backgrounds in neuroscience, psychology, and music performance for my Keystone project, and an exploration of music's effects on the mind, body, and brain. My project is titled The Influence of Self-Selected versus Pre-Selected Music on the Psychophysiological Person, and within it, I study the power of music to elicit or reduce specific emotional responses within a listener. In my experiment, I used both music that I selected, or Chopin's Raindrop Prelude, as well as a song or piece that each participant selected, in examining the efficacy of each condition or piece to reduce anxiety in the listener physiologically and psychologically. Finally, I also investigate this anxiety reduction on an individualized level, using interviews and questionnaires to explicate the fundamental faculty music has in influencing our emotions. For instance, do we imagine certain pictures or scenes while listening to music? Do we have specific memories tied to a particular piece or song? Do we even internalize the emotions that are expressed by it? The initial findings of my research were unanticipated. I found that although participants will report higher levels of psychologically perceived anxiety after listening to the Chopin piece I had selected versus their own musical selections, the physiological measures of their anxiety and arousal while listening to their own music are significantly higher. This lack of concordance between the psychologically perceived levels and the physiological indicators of stress suggests a lack of cognizance of our emotive and mood states, or that we may not always be as in tune with our emotions as we think we are. It is my hope that with this conclusion that this project can inspire future research along these lines in the integral and expanding fields of neuro and psychomusicology.
Okay, my name is Emily Powell, and I'm a student of the School of Hospitality Administration. So basically, going into my industry, I've always been very concerned about sustainability, and particularly how wasteful the hospitality industry is. And so basically, in trying to investigate sustainable practices in hotels, I did my literature review, and I basically found that there's pretty much nothing really between something like a really big, fat book that's really dense and convoluted with things like LEED standards, and something that looks kind of like this, where Mickey's telling you to turn off your lights. So things that are accessible but are not so dense and so kind of intimidating for hoteliers, things like that don't really exist. And so it was basically my aim in figuring out what to do with my Keystone project to fill the void, actually physically. So basically what I did was I wrote a book. So basically everything that I do in this book is I sort of detail, here are some of the main kind of standard and easier practices that hotels can implement to be as sustainable as possible, without getting too complicated. And they're all things that hopefully will encourage them to be sustainable through the use of things like models and statistics, and show them that it's not just helping the environment, but there are all sorts of energy costs savings that you can make. Water efficiency, energy use. You save a lot of money on those. People are afraid of how long the payback period is and things like that. So I kind of do that, and I sort of outline different areas of the hotel, and basically kind of just show them, here are some things you can do in things like waste management, water efficiency, energy efficiency, materials, things like that. And so that's my book.
Hello, my name's Yi. I'm a biology major, and for the past almost 2 years now, I've been working in Dr. Naya's lab, which focuses on molecular biology. So just, I guess, to give you a broad context to think about my project in, cardiovascular disease is the leading cause of death in the United States. And sort of to understand what's wrong, you have to understand what's normal first. So this is where fundamental basic biological research comes in. And our lab looks at gene regulation mediated by a family of transcription factors called MEF2. So just a brief refresher of intro to bio class. A transcription factor helps to regulate genes by essentially turning them on or off. So MEF2 family of transcription factors are involved in regulating genes involved in the heart as well as in the brain, but I will be focusing particularly on a transcription factor called MEF2A, which has been shown in previous studies to be extremely important in normal heart development and function. And so My project is titled Functional Characterization of a Novel Co-Regulator for MEF2-Dependent Transcription in Cardiac Muscle. That's a mouthful, so I like to also call it a tale of two transcription factors. So the first transcription factor is one that I've already introduced to you, MEF2A, which is important and critical in normal cardiac development and function. And so why is it critical? What genes is it specifically regulating in heart muscles? And a former student in our lab found that MEF2A regulates costamere genes. And so the costamere is this protein structure found in sort of striated muscle. So striated muscle involves skeletal and cardiac muscle. And it's been called the Achilles heel of these type of muscles. So it's very important. And MEF2A is regulating these proteins. So it's clearly important in maintaining the normal function of a muscle cell in the heart as well as in skeletal muscle. So then we delve a little bit deeper. How does MEF2A regulate these genes? And so we came up with a list of potential co-regulators, so what's working with MEF2A. And so the other transcription factor in this project is called EGR1. And EGR1 is a very interesting co-regulator to sort of investigate because it's also been implicated in cardiovascular pathologies. And so MEF2A is involved in troubles in the heart. EGR1 is also put them together, maybe they're working together. And I've actually shown that EGR1 is affecting MEF2A activity, so it's affecting the way that it's normally turning on and off genes. And sort of looking more into how this interaction between EGR1 and MEF2A affects cardiac muscle cells. So come on by and learn more about it.
So I believe our next student, Pat Reidy, is not here today. However, he has recorded something for us. And so if this works, I will proceed to play his recording.
Hi everyone, I'd like to talk to you today about the work I've been doing on sea urchin embryos. Bioelectrical gradients are important for biological processes, most notably neuron firing. And these bioelectrical gradients are set up by ion channels. And so what our lab did was perform a screen for drugs that inhibit ion channels and also produce a notable phenotype in the embryo. One of the drugs was called CON-A. Here you can see the phenotype that the embryos develop when treated with CON-A. We call this phenotype radialized, which suggests that dorsal ventral patterning is being perturbed. And so we wanted to further investigate how CON-A was perturbing dorsal ventral specification. Dorsal ventral specification is normally determined by expression of several genes, most notably nodal, which you can see in the middle on the bottom. Nodal turns on several other ventral genes, including BMP24, which will migrate to the dorsal ectoderm and signal there to determine the dorsal side of the embryo. And so, to characterize how CON-A perturbs dorsal ventral, we perform fluorescent in situ hybridizations, or FISH, on control and CON-A treated embryos at several time points while dorsal ventral is being specified. These experiments allow us to see where the targeted genes are being expressed and how they are changing over time. I won't go over each figure in detail, but the main conclusions that we can draw are that CON-A treatment promotes the radialization of ventral targets and loss of dorsal genes TBX23 and IRXA, which are downstream of BMP24. Interestingly, BMP24 expression persists with CON-A treatment, but does not properly signal to downstream targets, which is something that our lab will want to investigate further. Thank you very much, and I'd like to especially thank my PI, Cindy Bradham.
All right, you get a real person for this next one. Not that Pat's not a real person, but he's just not here. So 60 years ago, the American Law Institute created the Model Penal Code, which essentially created a standard for penal and criminal codes throughout all the states in the nation. And one of the things that they did was they defined levels of culpability with which you can commit a crime. They defined 4 different levels of culpability, and then of course there's blameless, which means that you didn't do anything wrong at all. And those different levels of culpability have different criteria that must be met in order for a person to be found to have acted with that level of culpability in committing the crime. But interestingly, it hasn't really been tested empirically if jurors or lay people can actually make those distinctions between levels of culpability, which is important because that's what they're asked to do on juries. So there's been a couple recent studies that where they've started to do these sorts of tests using scenarios that reflect these levels of culpability and asking jurors to either rate them or rank them, select levels of punishment for these different scenarios that the person should receive. And that's indicated that 2 of those different levels, knowing and reckless, that jurors might not necessarily be able to distinguish between those levels of culpability. So what I did is those surveys, they really looked at different scenarios. They looked at all of the knowing scenarios, all of the reckless scenarios. And I really wanted to know, are these findings actually occurring in courtrooms around the country? So what I did was I looked at the actual individual fact patterns, and using some other research related to intention and side effects and people's perceptions of morality, I was looking at to see if those individual fact patterns that were used, if there might be other factors that were influencing the jurors' decision process other than the definitions of culpability that legally they're supposed to consider. And so it seems that there might be some other factors that jurors are taking into consideration, which means that we may want to rethink how we're defining culpability, because if the legal definition doesn't match up with how people in their everyday lives perceive it to be, then that might be a problem, and we might need to consider reforming those definitions. So thank you.
Hi everyone, I'm Zach Herbert.
And I'm Ian Cohen.
And our team name for this project is called Microdiagnostics, and we're both studying mechanical engineering. So we were inspired originally by Professor Simon's Global Health section of our KHC sophomore course to pursue innovation in medical devices and global diagnostics. So we chose to focus on HIV, which affects a significant portion of the world's population, but also when diagnosed early can be controlled with medication.
We identified some of the shortcomings with current HIV rapid diagnostic devices and then built a better self-contained device. The device uses tiny needles to painlessly draw blood and runs a test for HIV with the results in under 15 minutes. We're really excited about the device that we've designed, and we hope that it can address these global health challenges.
Yep, and we have a lot of prototypes for you to check out, feel, play with, and so feel free to stop by our booth. Thank you.